So yeah, Saturdays this time of year are also, so I thank you for coming. They're important to me too.
I know.
It's the Ohio State football that unfortunately put a blackout on the Penn State Nittany Lions last Saturday. Ironically, I actually grew up 30 miles from State College, but I ended up going to Ohio State. My brother and sister are Penn State grads, but sorry to all you Penn State fans out there. So what I'm going to talk to you today about is introduce a little bit more about the plant initiative that we have here at Salk. And I think Joe's given a fantastic major part of the story, which is thinking about a new way of climate, the role that plants play, potentially as mitigating factors in climate, and the really major importance that understanding genomes has to potentially becoming or building systems and plants and understanding even natural plants, how they actually do something very positive for our climate. And I'm going to talk a little bit, or actually talk a fair amount about a natural substance called Cork. I'll introduce it in a little bit, and why this has become something that is of keen interest to us in the plant group here at the Salk Institute, and has formed a major part of our new initiative, and it has to do with climate. So this Harnessing Plants initiative actually started, in some sense, over 5 years ago, not so much with the scientists, actually. It actually came out of our board of trustees. We had one board member in particular, Howard Newman, who's now our major benefactor of the plant program. Howard runs a major investment firm in New York City and has, over the years, made a lot of money, actually, in the petrochemical industry, and felt a bit guilty about that. And Howard challenged us a few years ago during a talk I gave to the board that plants probably may have a solution to our problem, at least one of the solutions that we could think of. Because they do something that most people that think about the issue of climate change, and particularly the issue of carbon dioxide in our atmosphere, often think of CO2 as a toxic pollutant, but in fact, it's critically important. It actually, all the carbon atoms in your body, all living systems on Earth, actually come from CO2. So the idea with the plant initiative started with Howard, and then with my colleagues here at the Salk and the plant group, was to really reframe that issue and start to think about CO2 in a very different way. Think of it more as a fertilizer and the role that plants play in that, because they sit at the base of our global food chain. So even if you're a pure carnivore, you still depend on CO2 and plants to get all the carbon that goes into our bodies. So what is the issue with CO2? Well, it's a greenhouse gas. I'm sure everybody understands it, except for our federal government right now. And it does something very important for us, actually. It actually is a gas in the atmosphere, and so I don't know how many chemists or people that love chemistry or physics in the audience. Okay. And if the chemistry gets a little too intense, just tell me to slow down and simplify it. But what this molecule does in our atmosphere, along with others, but this is the major one, it sits up in our atmosphere, and sunlight comes in, it warms our Earth, which is important. And if we didn't have that atmosphere, that heat would re-escape, and we'd go through very hot days and very, very cold nights, like Mars, for instance. But what CO2 in our atmosphere does is it captures some of that infrared heat that comes off of the Earth, and rather than it's escaping back into deep space, it actually gets absorbed by these molecules of carbon dioxide, which are 2 oxygen atoms in a carbon atom, and it causes them to jiggle around, so they start moving and vibrating, dancing, and in that process, they give off some heat energy that reflects back on Earth. And so that actually moderates our temperature. So actually CO2 is very, very critical, particularly to mammalian life. It's what sustains life on Earth because it keeps us within this Goldilocks regime of temperature where we can actually survive. So it does something very important. The problem is that there's a lot of carbon buried in the Earth that my family and Joe's family over the centuries actually pulled out of the Earth and burned it. And that's also has been very important for our economies and our civilization, et cetera, but we've been burning too much of it. And what happens when you burn it? In the presence of oxygen, you make CO2, and so we're upsetting the natural equilibrium that the Earth has, and we've been putting too much of this in our atmosphere, and that's causing this steady climb in the global mean temperatures, which when you first look at it, it's 1, 2, 3 degrees predictions. You think that's not very much. But actually, if you calculate how much energy spread out over the globe, that excess energy into our ecosystems in the Earth, it's actually huge. And so it's causing lots and lots of problems, and we see it in our weather patterns. We're seeing it affecting, obviously, agriculture, et cetera, rising oceans, which I'll talk a little bit about. So it's a major, major issue of us dumping too much of this into our upper atmosphere. Of course, Al Gore, who's actually a friend of the Institute, we actually interact with Al quite a bit, has really campaigned over the last decade and a half on how do we reduce that through means of better renewable energy sources. And I have to say, he got a lot of flack early on for how financially, that it really wasn't something that we could do, and we certainly not something that we could do in a global way, particularly in developing countries. But I was just at a meeting getting trained by his climate reality group in Los Angeles about a month ago. And the cost of it is now down to the point where it's cheaper to do solar, wind, et cetera, than it is to actually mine coal, which is what my family has done historically in Pennsylvania. It's cheaper than mining coal, et cetera, out of the earth. So technology is really reducing how much CO2 we're putting up in the atmosphere, but we've already put a lot up. And so we need to also think about how do we draw that down and start to return the earth to equilibrium. So as I said, plants do this in a very miraculous way, I think. I grew up as a gardener in Western, I'll have a little side story about that in a bit. So including Mr. Bem, it was my great-grandmother and grandmother that also fueled my love of science, particularly getting my hands dirty in the soil. But plants do this wonderful thing. They actually capture that light energy from the sun, and they do this miraculous process called photosynthesis, where they draw in CO2. It's the carbon stock for all living things. And then with the energy of the sun, a little bit of water, they actually break this molecule up. They build all the carbon-based molecules that plants need to survive and prosper, and also to feed us. And in the process, they produce oxygen, which we also need to actually breathe. So they do this in a wonderful way without any kind of major technology, et cetera. And they potentially have the effect of drawing down that carbon dioxide in our atmosphere. Okay, so plants actually are this metabolic sink of solar energy historically. Here's coal from, I don't know if this is bituminous or anthracite coal. Pennsylvania's 2 states, actually. There's the Steeler fans on the west, the Eagles fans on the east. There's hard coal in the east and soft coal in the west. It's a crazy state. So in any event, plants actually build a lot of molecules, and that's my love of chemistry and linked up with biology historically, and particularly here at Salk, is really trying to understand all the complex chemistry that plants do, all the molecules they make. So they make things that we use for energy and for building materials. Cork, which I'm going to talk about in a minute. But they also, not lost, is that plants have historically also been a major, major source of pharmaceuticals. So for our own health, not only for nutrition, et cetera, but in treating disease, plants have and continue actually to be a major source of therapeutically important chemicals that for a chemist, any chemist in the audience will recognize these are very complex. And so while chemists have gotten very good at making molecules, they still don't rival what a plant can do with a little bit of water, a little bit of CO2, and a little bit of sun energy. Okay. So I'm going to introduce now this Harnessing Plants initiative and some of our major goals and why we think plants are a major way to think about as a major mitigating factor for climate change. So while we reduce, which is fantastic, and we become a more carbon neutral planet in terms of how much we emit into the atmosphere, we really can't solve the problem without also sequestering it. That is drawing down the CO2 that's in the atmosphere. And there's a lot of fantastic geoengineering and other engineering methodologies to do this, but unfortunately they actually right now take more energy to draw down CO2 than they actually pull out of the atmosphere, and they're not very scalable or distributive across the earth. But plants are, because you can now distribute those globally. They grow basically in every single ecosystem on Earth, from the very cold north to the tropical equatorial areas. So they are a very distributive solution to this problem. They are scalable as well. Modern agriculture is a prime example of this, where we've scaled native and natural plant systems to actually feed people. So it's a scalable problem, whether it's through genetic modification or advanced breeding methodologies or finding more and different nutritious plants. It is a scalable problem. And they have a track record. They've been doing it for hundreds of millions of years. Plants first moved from an aquatic existence probably 450 to 500 million years ago, based on the fossil record. And since then, they're the thing that really has turned the Earth, particularly the terrestrial Earth, into this fantastic living system that we all depend on. And they've been doing this, pulling this so-called toxic gas out of the atmosphere that entire time. Okay, so I said that they're naturally good at this, and one of the major goals, as you'll see in a minute, at the Institute, and my colleague Wolfgang Busch, who's here in the audience, who will speak next, is to really think about not only how they draw down that carbon, but how do we make it in a form that is more recalcitrant? All that means is that it stays, it doesn't get decomposed and returned back to the atmosphere because of the process of respiration, whether it's through us eating the plants or bacteria and fungi eating the plants or animals eating the plants. We want to take a portion of that and be able to sequester it or make it recalcitrant. And a major repository for that, and we'll make a case for it, and Wolfgang will do a much better job than I can, is to really focus a lot of attention now on the roots, where the soil is and where we can actually store that carbon. Okay? And as I've said, they've been doing this for millions of years. How powerful are they? Anybody recognize this plot? Yeah, go ahead. Yeah, what's it called?
The CO2 concentration in the atmosphere.
Yeah, but what's the name of the Keeling plot? So it's named after someone. Anyone know who that is? Yeah, where did he do this work? Yep, right down the road at Scripps Institute of Oceanography. So Charles Keeling back in the 50s was the one that began really monitoring, and first at the Mauna Loa Observatory in Hawaii, but now there's a globally distributed set of sensors that monitor in real time. You used to be able to go to the NOAA website and click on these and see the changes in real time and look at them historically. I don't know if that's changed. I know some things happened even this past week that you may not be able to access that data anymore. But Charles Keeling was the one that really started measuring this big time, and actually his house still sits down at Scripps Institute of Oceanography. So actually my group does a lot of work down there with my colleagues, and it's fun to actually link this up, and his son is continuing this work down at Scripps. So this is something that grew out of the science community here in San Diego. But what it shows, when he started measuring this in the late 50s, is that there's been this steady, well, not so steady, more exponential increase in the atmospheric concentration of CO2. And this, if I go back historically, and later today, if people want to talk about it, we have historical plots that go back further. This is really directly correlated with the rise of the Industrial Revolution. So you only see this exponential increase when we started burning more fossil fuels. But as a plant group, when you look at this, there's something really interesting. It's a little hard to see here, but it oscillates. So if we zoom in just over a single year or growing season, in fact, what you see is while the average is exponentially increasing, there's this oscillation. And this oscillation is perfectly timed to the plant growing season and the hemisphere in which you make the measurements. So this is actually measuring the northern hemisphere. And what you find is during the late winter and spring, when there's in colder areas where there's a thaw and all the plants that have died in the past season are really undergoing really rapid decomposition due to the fungi and bacteria made in the soil. And they respire and they emit CO2 just like we do. So you get this rise back up, which is a natural oscillation. But as plants start to grow and pick up sun energy, you see this drop. And this is not an insignificant drop. This is equivalent to about how much carbon we put up in the atmosphere every year from burning fossil fuels. So plants, if you look at this and think more about it, they have the potential to actually become a major mitigating factor in lowering that CO2 concentration because of this process of growth. Now the issue is we'd like to shave a little bit off of this yearly decomposition. So if we can just take a portion of what normally would be respired back into the atmosphere and bury it, for instance, in the soil, so that bugs don't eat it and respire, we now start to level this plot off. and potentially even draw it down over longer periods of time. Okay? So what are the three major goals of this harnessing plants stool? One, which you'll hear a lot about from Wolfgang, are deep roots. Go deeper into the soil, make them put forms of carbon in them that are more recalcitrant, so we can access a greater volume of soil and with more recalcitrant carbon. We want to identify molecules in plants that are recalcitrant. And I'll talk about this really interesting natural polymer called suberin, but most of you will recognize it as cork. It's the same thing that you find in wine bottles. And I'll make a case chemically why this is a very interesting molecule. And then I'll end, and I think one of our colleagues in the plant program who's a postdoc in Joanne Corey's lab, Carl Procco, I think he'll be here a little bit later to talk to you all. So Carl and I are also very interested in a very unusual group of plants that actually grow in our oceans called seagrasses and mangroves, and I'll end by talking about them and why we think that they're very interesting plants that haven't received a lot of attention with the kind of science we can do here at Salk historically, but now because of new technologies like Joe talked about, particularly in understanding genomes and epigenomes, we have now a blueprint for how these things have adapted to a marine environment, and I'll talk more about why they're interesting from a carbon sequestration standpoint. So let me just talk a little bit about suberin. This began also as a child back in Pennsylvania. My great-grandmother immigrated from France before the Depression. Her husband died, and she ended up having to feed my grandmother and my great-aunts during the Depression. So she did two things. She became a bootlegger, which is very common in Pennsylvania, and as we've learned over the last couple weeks, also in Maryland, with high school football players who then become judges on the Supreme Court. So she was a bootlegger, but she also had to feed her family. So she grew a garden all the time, and she was quite good at it, and that's where my love, when I was, even before I reached the age of 10, I really enjoyed gardening. But because I was a bit of a science geek, I also learned a lot more about compost bins, et cetera, and something that stuck with me that came back to me when we started thinking about this initiative was, this is not my compost bin. My dad wouldn't have allowed me to have one that big. But I remember putting corks into the compost bin. And while everything else would rot down and the volume would decrease substantially, these things never went away. But I knew that they were a natural substance. I thought at that time they were some form of wood. They're actually not. I'll talk more about it. They're more fatty acids, which Ellen talked about. So fatty acids are going to play a major role in this idea that we have about carbon in cork. So they never went away. They were recalcitrant. And some of you may recognize this if you ever put a cantaloupe rind in a compost heap. This is a vermicomposting heap with worms that are helping to assist the process. They rot and go away very rapidly, but one thing that's left behind, if you go back and sift through it, you'll see this outer rind with this netting. This is all suberin. So all plants actually make it. I'll talk more about it in a minute, but it appears to be a major recalcitrant form of carbon that plants make. And now the question is this something that could be beneficial for drawing down carbon? But also, what is its effect on soils? And I'll talk about that in a minute. Where do we get our major source of cork? Comes from the so-called bark of this tree. Anyone know what this tree is? Oh man, you are good. Yes. This is Quercus suber. It's the Mediterranean cork oak. So it's indigenous to the Mediterranean regions. They have massive plantations in Spain and Portugal. That's the major source of cork where they grow this. And they harvest it off of the outer portion of the tree. Anyone that's grown trees, if you would normally do this to a normal tree, that is girdle it, that is rip everything off, it would die. Cork oak is special. You don't actually injure the conducting elements that are so important near the surface of the tree because the cork oak through evolution is adapted to have this very thick outer layer. Anyone know why cork oaks probably make it? Think about Mediterranean region, or California even, huh? Drought is important. It does play a role in drought, but another major reason that it puts so much of it on the outer bark. What else? What happens around California usually this time? It fires. It's a fire retardant. If anyone's ever tried to burn a cork, Very hard. It's very waxy, fatty acids in it. It's hard to burn. This is a major protective layer of Mediterranean cork oak, so it's an adaptation. Even our live oaks here in California, if you go into the more foothills in the mountain regions where you see more live oaks, they actually have a layer of cork around them as well. And if you've ever gone and looked at those areas after one of our major fires, the oaks are doing just fine. So they resist that fire. So they do a lot of wonderful things for the plant. They also make it, what's not shown here, they make lots of it in roots. And I'll talk more about it, and I think Wolfgang will as well. Okay. So that's our source of cork oak. So we started to think about, could it be a good carbon sink in plant systems? And as I said, we'll make a case for it that all plants make it, and in particular, all plants make it in roots. It's a key adaptation of plant roots to surviving in very challenging ecosystems, both on land and in sea. So this has become the chemical or a focus of our initiative right now, and how to link all the kinds of science we do at Salk, all the way from chemistry to genetics and biology and now even ecology, to trying to understand how we can really tap into the beauty of this molecule called suberin, or as you are aware, also called cork. So it's a plant biopolymer. It's found in roots, both in the insides of roots, and then as roots age and get old, so-called secondary growth, it's found on the outside, much like the bark of the cork oak tree. It's a very carbon-rich substance. I'll show you in a minute a little bit more about it. It's actually made predominantly in these interesting cells on the outside of plants, whether it's above ground or below ground, called cork cells. Ironically, this was the first image of a cell. This gave rise to the whole cell-based theory of biology. The first cells ever observed were actually cork cells by Hooke. So these cells are very interesting. They produce lots of this suberin outside the cell, and then they go through a program of programmed death. So most cork cells are actually dead, they're often very empty, and that's why cork has that compressible nature to it. So they do a lot of things for the plant, and they insulate the plant. They regulate, in some sense, how the plant can interact with a very often toxic environment. And I'll show you some of the key roles they play in plants. And they seem to have this ability to retain carbon for long, long periods of time. I showed you the example, the compost heap, where suberin or cork actually resists degradation by organisms in our soil and on top of our soil. And I'll show you chemically why we think that's the case. And so the idea is that plants have been doing this for a long time and building up carbon-rich soils through a variety of different processes, but one major contributor we at least hypothesize is this natural plant polymer called suberin. Okay, so what we're doing here at the Institute is really trying to understand the genetic, the biochemical, and the chemical basis for how plants actually do this. And the goal then is not only to understand it, but to harness that and discover natural varieties of existing species that produce more of it, both in agriculture as well as in ecosystem services, but also potentially through the process of genetic modification, but again in a very tailored way, particularly in crops, can we increase the amount of suberin as a carbon sink? And then I'll also make a case why that also potentially will be very beneficial for growing healthier plants that can resist more extremes in our climate as they change. So the idea is that we can revitalize ecosystems, improve agriculture, and here is sort of what we call the 3-in-1 solution to suberin. One, I've already mentioned, the idea that they're a major carbon sink in our natural biological processes on Earth. But it turns out what we also know about suberin, particularly in roots, is that it greatly increases the plant's ability to withstand environmental stress. Someone mentioned drought. It turns out that plants that have increased suberin resist drought better than plants that have lower amounts of it. Ironically, paradoxically, they also resist flooding better. Two major issues with climate change are we're going to have more water in the atmosphere, but it's going to lead to more extreme droughts, but also more extreme flooding. And it can vary, as we see in California, from season to season. We'll go through periods of drought and then periods of a lot of water. So suberin, paradoxically, can resist both of those stresses on plants. They also are a natural resistance to pathogens. So instead of putting on insecticides and herbicides, et cetera, onto the plant, suberin actually helps resist those pathogens in the environment. And some of those pathogens can actually be animal or bacterial or fungal based, but also other plants, parasitic plants. They're actually known to help resist other plant pathogens. And so the idea then is by doing this, you get a better, more healthy ecosystem or agricultural system. You draw down carbon, and now that leads to better, improved plants for feeding people. So how do we get there? Well, we think across scales, from the very atomic level all the way up to the ecosystem and larger, to really think about it as what we term our ideal plant solution. And I'm a chemist, so I really wanted to know, first of all, chemically, what does suberin actually look like? I'll get into this structure, this complex structure in a minute, and how we actually derived it. But I wanted to understand, first of all, what it looked like. Second, how does it vary across plants? And with my colleagues at Salk, we're trying to understand how does the natural process of evolution change its chemical structure depending upon the requirements of a plant. And then also, why does it resist decomposition? And given my bias towards chemistry, I think chemistry can play a major role in trying to understand that. So what did we do? We did an MRI on Cork. This is a magnetic resonance imaging system that some of you had. I've done it to see if I still have a brain left. My brain is actually shrinking lately. But this is it's a magnetic resonance system, but it's actually called nuclear magnetic resonance. The term nuclear was taken out of it because people get scared, but there is no radioactivity associated with it. Well, we can do the same thing. In fact, we did it before the MRIs in the lab and used a thing called nuclear magnetic resonance, where in this dewar of liquid helium, there's a little channel, there's a very strong magnet, and also a radio frequency generator that makes radio waves. And we stick the molecules down into this very cold environment. We blast them with a magnetic field and with radio waves. And then we can detect fingerprints of chemicals, and we can actually put together, like pieces on a puzzle, the chemical structures of complex molecules. And that's what we actually did with collaborators at Brandeis University near Boston, to actually begin to understand what is the structure of this suberin molecule. Okay? So I'm not going to go into this, but you get these, what I call fingerprints, and they're complex, but you can actually begin to read these off. There's lots of information in these about what is the atom type, who is that atom type connected to, what are the chemical bonds that make up those atoms. And we can also, by using different timing regimes, we can actually even measure distances, okay? So with this, we can put together, just like pieces on a puzzle, what these complex molecules look like, and we can tell also how big they are or how small they are. And so we get these fingerprints. This is called a 1D sample. The previous one was a 2D. We can go 3D, we can go 4D. With better and better computers, you can go further and further and further. And we actually looked at commercial cork that has been processed and put into wine bottles. But actually, we have a stand of Mediterranean cork oak up in the botanical gardens up in Encinitas, who allowed us to go up there and actually drill into some of the outer bark and collect native cork oak that you would harvest. We put it in this machine, and they're identical. So you get some small differences when you process it. It's a little pure, but for all intent and purposes, the fingerprints are identical. But here's the thing that really made us very, very excited. So my colleague at Brandeis, before he went there, he was at Iowa State University for a number of years. And because Iowa State is a land-grant school that's really interested in agriculture, part of his time was spent sticking soil into that NMR machine, doing magnetic resonance imaging on soils. And when we did these first experiments now a couple years ago, he had previously looked at soil carbon or peat human, et cetera. And he's a German guy. You'll get a much more excitable German with Wolfgang. My colleague Klaus, a little bit more reserved, but not when he presented this result. He got very excited because he said the soil carbon fingerprint that we previously determined is matching identically to what we see in suberin. So this led to, again, a major hypothesis for us, which is the suberin that we find in cork, that's found in all plants, particularly in roots, may be one, not the only, but maybe the major source of carbon found in very carbon-rich soils. We also know that these carbon-rich soils are more fertile. They grow plants better. Plants are more able to sustain a drought and flooding, et cetera, because of the nature of those carbon-rich soils. So it appears that not only is it a sequestered form of carbon in our soils, but it also leads to more fertile, healthier soils. And so again, all these little pieces of the puzzle get put together where this becomes now one of the areas that we're focusing quite a bit of attention on in the plant initiative. Okay, so this is a little bit closer. These are fatty acids. So these are just like the fatty acids you and I make. They're long. They're about 30 carbons in length. And that's actually really important because the dimensions of this exactly match what you see if you stick this suberin in an electron microscope. Just like tree rings, you see these alternating light and dark layers. We can measure these with a high degree of accuracy, both in the electron microscope, but also in the NMR, the nuclear magnetic resonance machine. And what you find is that you have these fatty acid layers, and then you have what are known as the polyphenol layer that are stitched together, and they're all stitched together with glycerol, turns out. You make things like triglycerides or diglycerides. They're all stitched together, and then they form these layers. So this is just one piece with the fatty acid layer and the two outer polyphenolic layers. And then you would build another one on top of that. And you would form layer after layer depending upon the growth season in the plant. And they form this very interesting chemically complex barrier in plants that can regulate lots of things. It turns out that this is the part, the polyphenol layer, is the part that's more degradable. It's the part that bugs actually will preferentially use as an energy source or as food. But what they don't seem to be able to access very well is this fatty acid layer. So this is a lot like hydrocarbon oils and other things that we get out of the petrochemical industry that bugs are not very good at accessing as an energy store. And so this is the part that seems to be left behind in carbon-rich soils. Okay. So we're very interested in now moving from this basic understanding of the chemical structure to what it means in terms of plant growth and resilience, but also in terms of carbon storage. So I think Joe has introduced this. Wolfgang will also talk more about it. Salk works on these so-called model systems, as Joe said, but they're actually real plants. He talked about the Arabidopsis project, where you can actually look at the same species, but growing and evolving in very, very different climate zones and environments, and link changes in the evolution of the genome to how it can survive these sessile organisms in all these different environments. And then Wolfgang is going to talk about Lotus, I believe, which is a fantastic system that also is a bit of a different plant. It's a legume, so it fixes nitrogen and it does some things that Arabidopsis doesn't do. But again, you can look at it in its native context across the Japanese Isles from the cold north all the way to the tropical south. So single species, but adapted to widely varying climate zones. And then my sort of guilty pleasure right now is shown here. This is a species of marine seagrass called Zostera marina, or eelgrass. And in fact, this is the thing that grows in our local oceans here off the coast. And I'll talk more about it. It's a real plant. It's not algae. It's not kelp. It's not something like that. It's actually a real plant with roots that bury into the ocean sediment. And they're found globally across all oceans, from the tropical areas all the way up into the frozen Arctic, actually. So you have these plants that are capable of evolving and living and exploiting very, very different ecosystems. And I'll talk more about it in terms of carbon sequestration in a minute. So our solution really is to really look at the terrestrial system and think about how plants can impact the emitted CO2 we put into the atmosphere, and also now start to tackle this group of plants that have been historically very understudied, these marine systems, particularly seagrass, but also to some extent in the subtropical and tropical areas, mangroves. And I'll talk more about them in a minute. So they're really an unexpected ally, we think, in this global carbon cycle initiative of how plants draw down carbon. So I'll introduce a little bit about them. They're major ecosystems for our economies, particularly fisheries. But it turns out they also capture carbon probably better than any plant-based ecosystem on Earth. It's estimated that upwards of 30 times greater carbon storage capabilities in these marine systems than on land. And there's some specific reasons having to do with how the plant evolves, but also the chemical environment that they're found in. The problem, of course, is through human development and other things, we've lost a tremendous number of these ecosystems, which has resulted in, I'll show you a prime example of that in a minute, major impacts on our environments and our economies based on fisheries, et cetera. And they're very important for fisheries. It's where all of our fish, shellfish, actually breed, but they also very effective at removing pathogens out of the oceans. So we've had, for instance, over the last several years, it's particularly in the news now in Florida, a few years ago here on our west coast, these toxic phytoplankton blooms. These plant-based systems are very good at removing a lot of those out of the ecosystem. So obviously, as we lose those, we have a major impact on the health and well-being of our ocean environment. As I said before, they're globally distributed, so they're basically found everywhere in our coastal areas. They grow anywhere from subtidal areas where part of the day they're above the water to very deep areas. They've actually found seagrasses all the way up to 90 meters in depth, which is a bit confounding to us as plant biologists because they're very deep. There's not a lot of light available, but they're still able to capture enough light and actually survive in those environments. They have, in some cases, very deep roots. This is something that I'm learning a lot from Wolfgang. It's fantastic that he's here at the Salk now. And in some of the cases, one of their adaptations to living in this salty environment is suberization. So suberin in roots is a very protective mechanism, not only for drought and flooding and pathogens, but it also helps plants resist growing in very salt-rich areas. And that's a key adaptation for not only seagrasses, but also mangroves. And so, and if this is the thing that captures a lot of carbon, when we know these ecosystems bury a lot, it's something that we're keen to really study. Problem is over the last 2 decades, going back to 1990, we've lost on average about 7% of these a year. So since 1990, we've lost more than half of our seagrass beds globally, and that only accounts for 1990 to the present. It doesn't even account for going way back. And that's due to a number of things, mostly early on and through human development. But as climate change sort of ramps up and the sea levels are rising and the salinity of our oceans and temperatures are changing, it's a major impact on these ecosystems. And in fact, this year we've recorded the highest temperatures off of the La Jolla, the Scripps Pier ever in our oceans. I think 79 degrees Fahrenheit, which is unprecedented in our oceans. Turns out our major seagrass here, that Zostera or eelgrass, when the temperature starts rising much above about 72 degrees Fahrenheit, they're under significant pressure. When you get above 75, 76, you're going to lose those seagrass beds. So in fact, we've been looking in some of our local bays, Mission Bay and San Diego Bay, at some of the natural beds with Carl Procco and Joanne's group. And we think that this fall we're going to have a major die-off of our seagrass beds because of the rising ocean temperatures. So part of this initiative is to really start to understand how plants, in terms of their natural variation, how they can resist higher and higher temperatures so that we can actually partner, as I'll show you in a minute, with restoration efforts to begin to restore in a very informed way varieties that are going to be better able to tolerate the changing sea temperatures, salinity, et cetera. As I said, they're obviously a key breeding ground. They're a major role in controlling erosion. We're working with a group in Louisiana where they're very interested in these systems to actually build land rather than lose land through storms. They do reduce ocean acidification because the CO2 dissolves in salt water. It's alkaline, so it turns into bicarbonate, just like the bicarbonate you buy in the store. And that's the form of carbon that the seagrasses actually absorb. When they do that, so when you dissolve CO2 in ocean water, it makes the ocean more acidic, but when the plant grabs it and takes it out of the ocean, it makes it more alkaline. So in fact, if anyone's interested, I can point you to some really fantastic videos where you can measure the pH of the marine water in an enclosed aquarium system before you turn the lights on and after, and you get almost a 1 pH unit change in the pH of the water because it's so efficient at grabbing all this CO2-based carbon out of the water. They store and they grab a lot of carbon not only in the ocean, but also runoff from rivers, et cetera. Because they're floating around in the water, they actually sediment debris very effectively. and then they bury it in the seabed. And because the seabed accretes, that is, it gets thicker much more quickly than on land, you build up these very, very carbon-rich, deep sediments. And so people have been probing those, and there are some that are, through carbon-14 dating, up to 6,000 years old and 10 meters deep in coastal deposits. So you can bury a lot of carbon in this environment, and it stays there because there's very little oxygen. Okay? So what can we do? Well, only 2 years ago, the first genome came out of a seagrass, which turns out that's the species we're most interested in. So this now provides us with the blueprints for understanding at a molecular level how these systems are adapting and how they're storing carbon. And so this is a wonderful rule book that we can actually tap into. It's also an opportunity for us because there has been, as I said before, historically very little work done by the kind of science we do here at Salk on these systems. And so this makes for a very exciting area of science to probe, but it also is now forcing us to interact with people outside of our own fields, ecologists and policymakers in industry. We meet with a coastal commission. We meet with, I'll show you in a minute, a group of PADI certified divers who are interested as citizen scientists in participating in this. And Carl Procco, who'll be here later, is going to talk about how he's now bringing in high schools and undergraduate students into this program to really become part of this scientific initiative. So we're building these collaborations. They're important, as I said, in fisheries, so you can actually do the economics. You can make a very strong case why restoring these systems is a major boom to economies, particularly in developing countries where fisheries continue to play a very important role. As I said, they reduce pathogens, so we're starting to understand how they contribute to the hygiene of the oceans. This is one that Al Gore sent me back in the spring. This is what's called a carbon bomb. This is a seagrass bed of a species similar to ours that grows here in California that experienced a heat wave, just like we're going to experience this fall, where a large number of these seagrass beds that are very extensive actually did a massive rapid die-off. And when they did, all this carbon that was stored now gets exposed to oxygen, and it caused what's known as a carbon bomb. So there was this huge release now of all that carbon. So they store carbon, but we have to maintain them because if you lose them, you can actually release lots of carbon dioxide very, very quickly into the atmosphere. This is in the children's pool down in here in La Jolla. There are 3 major seagrasses that grow in our oceans, and the one whose genome has been done is Astera marina. And so we've now begun monitoring these and working with the military as well as local NGO groups that are very interested in restoring these systems. And again, we have a blueprint for the genome, and we now are getting ready with Joe's assistance to look at other seagrasses, different species, and really look at their genomes as well to try to understand how this very interesting group of plants are adapting their genomes to survive in these environments. So we can learn a lot about their biochemistry by understanding the genomes. They have these wide geographical zones, so for Astera alone, you can see just like with Arabidopsis or lotus, which I showed previously, it grows in a wide range of different latitudes and longitudes, as well as like day length cycles and temperatures. So by sampling how the genome of this single species is changing in all these environments, we can start to do predictions as to what variety is going to be better grown. Let's say up in the Pacific Northwest, as the average temperatures of the ocean there increase, maybe a variety of the same species that's actually growing off of Baja can be now used for restoration efforts up there. And there's a lot of restorations going on of all the seagrasses globally. The unfortunate thing is the way they do restoration now is you grow a bunch of plants and you put back what grew there before. In the past, that was probably a good thing if we destroyed the bed, but if the beds are being lost through climate change, you put back the same thing that grew there and you wait a few years, the experiment's going to fail, and we don't have time to do that. So the idea is to now inform these groups with genetic information, biochemical information, to better inform them that no, don't put back that variety, put back this other variety, because it's going to have a much higher likelihood of being a success. Okay, so I talked about that. I'm almost done. And you'll hear more from Carl. I won't steal his thunder. He's very, very interested in outreach, and he's been doing a lot for this program of sustainability. So I think he's going to talk about how high schools can get involved, not only in in Arabidopsis and understanding it, but also the seagrass program that we're building here at the Institute. Let me just end with that. Let me go. So we have a wonderful team, I think, not only with collaborators, but here at the Salk with very complementary areas of expertise that you've heard from Joe and you'll hear from Wolfgang in a minute. So we all complement each other to really understand how plants do this amazing thing of grabbing CO2 out of the atmosphere and building lots of interesting and cool molecules that are important for all life on Earth. And I'll just end with Jonas's famous quote. Thank you. Yeah.
When you were showing the Keeling curve and the annual variation, you said that one of the goals was to kind of reduce that initial uptick. I was wondering if you had any insights into if you thought that would affect the overall food chain if we're not putting that back in.
Right.
Because the bugs and the detritus food
Right.
have that energy.
Yeah, it's a great question. A large portion of that, the capture and the uptick, is agriculturally based because of the massive way we do agriculture now. What we would like to do is return it to more of a natural system, but to do it also in crop plants. So the idea is, could we be growing crops that are burying more carbon in the soil? And these are things that we really have to quantify and study to see what effect it might have on the food chain. We would make a case that at least based on what we know about suberin, that it actually might improve the food chain because the plants that you would grow in a field, wherever it's at, that are experiencing, farmers have major losses every year, depending upon what's happening, that they're going to be able to better resist that. So the hope is that by having crops that have more suberin content, for instance, in their roots, that the food system will be more sustainable than it would have been otherwise. And in the process, we get this extra thing, which is we draw down some of that carbon out of the atmosphere. But they have to be measured and then modeled in a major way. And it's the thing that we have some modeling here at the Institute, but we lack, and that's why forming partnerships and also bringing in new people into this initiative become very important so that we can directly address that question. Yeah.
You mentioned that the suberin was related to improvements in soil fertility.
Yeah.
What specifically is that related to? Do they increase the cation exchange capacity? What's
Yeah. Ooh, I'm sorry, Joe. Yeah, great, fantastic question. They do a lot of things. They hold on to water more. They also do like a human. Human is a kind of an amorphous word for soil carbon. They do very similar things because the molecules are not only fat-like and oily, but they also have charges at specific locations on them. And those charges are very important because they can hold on to water better, so they can act as a molecular sponge for water, but also key nutrients, particularly cations in the soil are a thing. And then what happens, what you find, just like with, there's a lot of interest in biochar, which is to some extent similar to this. You burn in a very oxygen poor environment, you burn plant biomass and you turn it into charcoal. You put it in the soil, plants seem to do better. And one of the major ways they do this, and a lot of chemistry has been done on those systems, they hold on to these nutrients and then they just slowly release them to the plant rather than boom, a whole bunch at a time. So it's probably a combination of water retention and the regulated release of important nutrients for the plant, whether they're cations or plant hormones that plants secrete, et cetera.
I was going to do one quick comment. When I came in 1973, I became a scuba diver.
Yeah.
And the cove area was stripped bare. And that was because of overfishing of the lobster and anything that was around there. And so it was established, we were scientist undergrads who really didn't know what we're doing, but we tried to create transit lines and establish what was down there as a baseline. And so, that was 1973. I saw an amazing improvement
Oh, yeah.
into what the flora and fauna are here. So, it'll be interesting to hear students can get involved and really have an impact. Even back then, I had no idea what I was doing, but we got it started.
Yep.
So
Yeah, it's really science out in out in the field or
Yeah, and it
in the
was-
ocean
And
or
those
whatever.
seagrasses were not
Yeah.
as rich as they are now.
No.
It's sad to hear that they're gonna die off.
Question over here.
Yeah.
Yes, and you might have said this, but I'm so overwhelmed with all I'm hearing, many questions. Are there other countries that are studying this?
Oh, yeah.
Research?
Yeah, so seagrass has been and by the way, I can share my talk with anyone that wants it, so you can.
We will.
Okay.
Yeah.
Oh, yeah, and it's been recorded too.
You've been recorded, but
Yeah.
also
Yeah.
you'll make your
The
slides
slides.
available.
Yes,
That'd be
and then
great.
any information, you can always contact me and share it. Yeah, there's particularly since the genome's been done, it's been in the New York Times, The Guardian, I showed you. There's a lot of interest globally in these systems. And actually, the genome was done by a consortium of people. The actual work to actually sequence it was done at the Joint Genome Research Institute up in Walnut Creek in California. But it was people from Europe, United States, South America, Central America, some in Africa, et cetera. So it's a global group of people. And so this is not something we can do by ourselves. We have to really partner. And the idea is not to be competitive with this kind of science, but to actually be integrative and really work with people. But I also think, you all being high school teachers, and I, growing up in middle school, where I grew up, we didn't, there was no science lab or anything. The only thing I could learn was going out in nature or reading a book. But I think nature sometimes is the best teacher. Books are great, visiting labs are fantastic, but there's a lot of science that you can learn by just getting out there. And I think that's arguably why plants are really interesting, because they're things that you can do in your home and you can do in the lab and you can do in your school. So these are great systems for learning, but also making an impact and actually doing something that's going to change, we hope, the world actually for the better. Yeah.
If you could make a prediction,
Yeah.
how, like as a timeline, when do
Yeah.
you think you would be able to have like these mangroves that are more temperature resistant, or these crops that make more suberin, and you're actually disseminating it and it's going global?
Maybe, and Wolfgang, this is something we were always continually now arguing about in our initiative. The goal would be within a couple years we have the first plants, probably in crop systems. We have some collaborations that are likely to go through with some companies that are doing a lot and have actually experimental plots globally where things can be tested. With seagrass, we're hoping, with the help of others, we're already interacting with NGOs that are doing a lot of restoration. So, but what needs to be done now is to take what we know here at the Salk about temperature adaptation, light adaptation, et cetera, look at the genomes, but across many genomes of for instance, Zostera that have been plucked from different locations, and then partner up with these restoration efforts. So in fact, we're part of our group are going to go to Louisiana in early November to work with a group that are interested in salt marsh restoration in Louisiana. And there we hope not to only get this work going, but identify new species of seagrass that we should also be focusing on that are important in the warmer areas of our oceans, et cetera. So the hope is that within 5 years that we're doing things, but that really is going to depend on the partnerships that we form.
Okay, we have to take our break.